Hi everyone. We will present topic 10, the beta fatty acid oxidation. In this video, we will study about beta oxidation in which free fatty acids are converted to acetyl-CoA. The whole process of beta oxidation is divided into three phases: A. Transport from adipose tissue to target cells. B. Entry into cytoplasm and mitochondria. C. Oxidative catabolism inside the mitochondria matrix. Firstly, we will talk about the transport of free fatty acid from adipose tissue to target cells. Human body contains adipose tissue that consists of triglycerides. The triglycerides contain fatty acids and glycerol molecule. Next, the fatty acid will be distributed all over body before they enter the blood circulation. Then, the free fatty acid will enter the bloodstream and reach any cell except RBC and nerve tissue to metabolize the free fatty acid. RBC and nerve tissue unable to metabolize the fatty acid because they lack mitochondria, as it is essential for fatty acid metabolism. After that, the free fatty acid will first enter the cytoplasm of the cell through the fatty acid transporter. They require fatty acid transporter since they have a net negative charge that disallows them to pass the cell membrane. Afterward, the free fatty acids enter the matrix of mitochondria after crossing the outer and inner mitochondria membrane. This matrix of the mitochondria contains all the enzymes that are required for beta oxidation of fatty acid. It helps to balance the enzymes for Krebs cycles and electron transport chain. Next, we move to activation and transport inside target cell mitochondria. The free fatty acid then freely enters the outer mitochondria membrane, which contains porins, which is very permeable to many substances including acyl-CoA. However, inner mitochondria membrane is not permeable as it requires carnitine shuttle, a special mechanism to transport the acyl-CoA through the inner mitochondria membrane into matrix. The carnitine shuttle contains many enzymes, and one of them is the carnitine palmitoyltransferase 1, located in the outer mitochondria membrane. It will add a molecule of carnitine to acyl-CoA. In this reaction, the CoA enzyme of acyl-CoA is released back into the cytoplasm. In the intermembrane space, a molecule of acyl-carnitine is generated. In the next step, the enzyme carnitine acylcarnitine translocase, which is located on the inner mitochondria membrane, will transport this acylcarnitine into the matrix of mitochondria. The free fatty acid must be converted into its active form, that is acyl-CoA, to be metabolized inside the mitochondria matrix. Once activated, the free fatty acid is transported to mitochondria. The acylcarnitine molecule must be degenerated by the enzyme carnitine palmitoyltransferase 2. CoA enzyme added and carnitine is regenerated back and transported again into the intermembrane space. Now we move to Part C, the oxidative catabolism, inside mitochondria matrix. In this compound, the word fatty in the name fatty acetyl-CoA can be replaced depending upon the length of the side chain that is attached to the functional group. For example, palmitoyl-CoA, acetyl-CoA and myristoyl-CoA. As the fatty acetyl-CoA with 16-carbon compound enter the matrix of the mitochondria by the carnitine shuttle mechanism, using 4 enzymes. 2 carbon atoms will be removed from the parent fatty acid, lead to generation of 2 molecule. The 2 molecule, one is acetyl-CoA, and the second molecule is a new fatty acetyl-CoA, which is 2 carbon shorter than the original parent fatty acid, will become 14 carbon compounds called myristoyl-CoA. As for 16-carbon fatty acid, the palmitoyl-CoA, to become broken down to 8-molecule acetyl-CoA, it must undergo 7 cycles of beta oxidation. During these 7 cycles of beta oxidation, the 4-carbon side chain left, which is broken down into 2-molecule acetyl-CoA. After 7 cycles of beta oxidation, we have 8-molecule acetyl-CoA, we have 7 molecules of NAD, and we have 7 molecules of FOD2. This reaction takes place in the mitochondria and the enzymes from Krebs cycle and electron transport chain will be utilized. So this is how through the process of beta oxidation in the mitochondrial matrix, a 16-carbon fatty acid, the palmitoyl-CoA is broken down completely into 8 molecules of acetyl-CoA. The process begins with dehydrogenation. Double bond formed between the beta and alpha carbon since hydrogen atoms removed. This reaction is catalyzed by enzymes acyl-CoA dehydrogenase and produces a compound named transinyl-CoA. The hydrogen atoms removed are donated to electron acceptor FAD, which is reduced to FOD2. The resultant molecule is called transinyl-CoA because it is in a The second reaction of the beta oxidation in the mitochondrial matrix process is hydration reaction, which means the addition of water. Water is added around the alpha and beta carbons. A hydroxyl group is added to the beta carbon and a hydrogen atom around the alpha carbon. This reaction is catalyzed by enzyme acyl-CoA hydratase. The compound produced is called hydroxyacyl-CoA. The process continues with a third reaction, which involves a dehydrogenation reaction again. Double bond formed between the carbon and oxygen atom after removal of hydrogen from the beta carbon of hydroxyacyl-CoA. The hydrogen atoms removed are donated to electron acceptor NAD, which is reduced to NAD. This reaction is catalyzed by enzymes hydroxyacyl-CoA dehydrogenase and produces a compound named ketoacyl-CoA. The last reaction of this beta oxidation in the mitochondrial matrix is called teolysis, which means a cleavage reaction. During this reaction, the molecule of coenzyme also used and it is catalyzed by enzyme thiolase. The ketoacyl-CoA is then broken down into 2 molecules. The first is a new fatty acid which has a side chain of 14 carbons called myristoyl-CoA, and the second molecule is acetyl-CoA. The first 3 reactions involved in the series of the beta oxidation is just for the preparation of fatty acid to a more balanced and symmetric molecule which can be broken down into 2 molecules. The myristoyl-CoA can be produced again, enters the series of beta oxidation, and it is completely utilized to form 8 molecules of acetyl-CoA through similar reactions. Energy yield of beta oxidation. For the calculation of energy, 8 molecules of acetyl-CoA are fed to the Krebs cycle. The 7 molecules of NADH and FADH2 are fed to the electron transport chain to produce energy. 1 molecule acetyl-CoA gets 10 ATP through the Krebs cycle. Meanwhile, after considering the real situation, 1 NADH equal to 2.5 ATP and 1 molecule of FADH2 equal to 1.5 ATP. The energy yield of beta oxidation of acetyl-CoA will give 80 ATP through the Krebs cycle. 7 molecules of NADH will give 17.5 ATP through electron transport chain, and similarly 7 molecules of FADH2 will give 10.5 ATP through the electron transport chain. In total we have 108 ATP, but it is crucial to deduct 2 ATP which has been used to utilize the activation of palmitoyl-CoA. The energy yield through the 16-carbon fatty acid is 106 ATP per molecule of palmitoyl-CoA. So, that is all the process of beta oxidation. Thank you for watching it.